In September 1998, an unusual cluster of illnesses began appearing among pig farmers in the Malaysian state of Perak. Men who worked closely with swine started developing fevers, headaches, and rapidly declining neurological function. Within days of symptom onset, previously healthy individuals would fall into comas. The illness was devastating, and medical professionals initially struggled to understand what they were facing. The Malaysian health authorities first suspected Japanese encephalitis, a mosquito-borne viral disease known to affect the region. This misdiagnosis delayed the implementation of appropriate control measures and allowed the outbreak to continue spreading. By March 1999, the outbreak had reached the neighboring state of Negri Sembilan, and an alarming pattern had emerged across multiple pig farms. The affected individuals were predominantly adult men who had direct contact with pigs, suggesting an animal reservoir was involved. The situation took an international turn when 11 workers at slaughterhouses in Singapore developed similar symptoms after handling pigs imported from the affected Malaysian farms. Scientists working urgently to identify the causative agent made a startling discovery. This was not Japanese encephalitis at all, but an entirely new virus. The pathogen was isolated from both infected patients and pigs in March 1999, and researchers confirmed through cross-neutralization studies that while it shared similarities with Hendra virus, it was a distinct member of the Paramyxoviridae family. The virus was named after Kampung Sungai Nipah, the Malaysian village where it was first identified. The scale of this initial outbreak was catastrophic. Between September 1998 and May 1999, Malaysia reported 265 confirmed cases of Nipah virus infection, with 105 deaths, resulting in a case fatality rate of approximately 38.5%. Singapore recorded 11 cases among abattoir workers, with 1 death. The Malaysian government responded with drastic measures. They implemented nationwide testing of pigs, established quarantine zones around affected farms, restricted animal movement from infected areas, and ultimately ordered the mass culling of over 1 million pigs from any farm with confirmed infections. These aggressive interventions, combined with thorough cleaning and disinfection protocols, finally brought the outbreak to an end by mid-1999. No new cases have been reported in Malaysia or Singapore since that initial outbreak, but the virus had announced its presence to the world in dramatic fashion. Chapter 2: Understanding the virus and its natural reservoir. Nipah virus belongs to the Paramyxoviridae family, which includes measles, mumps, and respiratory syncytial virus, but Nipah and its close relative Hendra virus form a distinct genus called Henipavirus. The virus possesses a single-stranded, non-segmented, negative-sense RNA genome encapsulated by envelope proteins. 2 critical surface proteins enable the virus to infect cells: the G-glycoprotein, which binds to cellular receptors, and the F-fusion protein, which allows the viral envelope to merge with host cell membranes. Scientists discovered that Nipah virus binds specifically to Ephrin B2 receptors found on epithelial cells and neurons. This receptor binding triggers conformational changes in the viral proteins that facilitate fusion and entry into host cells. Researchers identified several key amino acid residues in the G protein, including E533, W504, E505, N557, Q530, T531, and A532, that play essential roles in receptor binding and promoting viral fusion. The virus also produces proteins designated C, V, and W from its P gene, and these proteins help the virus evade the host immune system, contributing significantly to its pathogenicity. When the initial outbreak occurred, scientists already suspected that fruit bats might serve as the natural reservoir based on knowledge about the related Hendra virus. This hypothesis proved correct. Subsequent investigations detected Nipah virus RNA and neutralizing antibodies in the urine, saliva, serum, and organ tissues of multiple species of pteropus fruit bats, commonly known as flying foxes, throughout Asia. These pteropid bats are distributed across Oceania, South and Southeast Asia, and evidence suggests Nipah-virus-related antibodies exist in African fruit bats of the genus Eidolon as well. Remarkably, infection with Nipah virus does not appear to cause disease in fruit bats. The bats carry and shed the virus without suffering illness, making them perfect reservoir hosts. Studies have documented the presence of Nipah virus in bat populations in countries that have never reported human cases, indicating the geographic range of the virus may be far more extensive than current human infections suggest. The virus persists in bat colonies year-round, creating ongoing opportunities for spillover events when humans or intermediate animal hosts come into contact with bat secretions. Chapter 3: Clinical Manifestations and Pathology. The incubation period for Nipah virus infection ranges from 4 to 14 days in most cases, though rare instances of incubation up to 45 days have been documented. Some individuals may experience asymptomatic infections, but most develop acute illness characterized initially by fever, headache, muscle pain, chills, fatigue, drowsiness, and dizziness. For many patients, the disease rapidly progresses to involve the central nervous system, causing altered consciousness, confusion, drowsiness, and encephalitis. During the original Malaysian outbreak, neurological symptoms dominated the clinical picture. Patients developed signs including areflexia, hypotonia, segmental myoclonus, and gaze palsy. The mean time from fever onset to hospitalization was only 3.3 days, and the mean time from fever onset to death was just 9.5 days. Many patients deteriorated so rapidly that they progressed from initial symptoms to coma within 24 to 48 hours. This terrifying speed of disease progression became a hallmark of Nipah virus infection. Subsequent outbreaks in Bangladesh and India revealed a different clinical presentation. Patients infected with the Bangladesh strain of Nipah virus frequently developed severe respiratory symptoms, including cough, difficulty breathing, and acute respiratory distress syndrome, in addition to neurological complications. The respiratory involvement can be so severe that patients require mechanical ventilation. Pathological examination of fatal cases reveals widespread systemic damage. The virus causes vasculitis of small blood vessels throughout multiple organs, leading to thrombotic occlusion and microinfarction. In the brain, necrotic plaques appear in the parenchyma, and occasional syncytial or multinucleated giant endothelial cells are observed. The virus demonstrates neurotropic properties, targeting neurons and causing inflammation and swelling of the brain. Multiple organs beyond the brain and lungs can be affected, and the extensive vascular damage contributes significantly to mortality. The case fatality rate varies depending on the outbreak and viral strain, ranging from 40 to 90%. The Bangladesh strain appears more lethal than the Malaysian strain, with an overall case fatality rate of 71.7% across all documented outbreaks in Bangladesh through September 2025. Animal studies using African green monkeys infected with both strains confirmed increased lethality and more severe pulmonary pathology in the Bangladesh strain group, suggesting viral virulence factors contribute directly to the higher mortality. Among those fortunate enough to survive, most make a full recovery, but approximately 1 in 5 survivors experience long-term neurological sequelae. These persistent neurological conditions can significantly impact quality of life and serve as a sobering reminder that even surviving Nipah virus infection can leave lasting consequences. Chapter 4: Transmission Pathways and Outbreak Patterns. The transmission dynamics of Nipah virus have evolved considerably since the initial Malaysian outbreak. In Malaysia and Singapore, pigs served as an intermediate host, amplifying the virus and transmitting it to humans through close contact. The virus could be isolated from the nose and oropharynx of infected pigs, and pig farmers and slaughterhouse workers faced the highest risk. This pig-mediated transmission has not recurred since 1999 in those countries. Beginning in 2001, a distinctly different pattern emerged in Bangladesh and India. Investigations revealed that many patients had consumed raw date palm sap, a sweet beverage collected from date palm trees during the winter months from December to February. Fruit bats visit these trees at night to drink the sap, and in the process, contaminate the collecting pots with their saliva, urine, and excreta. Humans who consume this contaminated raw sap can become infected with Nipah virus. Studies documented that almost half of primary cases in Bangladesh have been linked to date palm sap consumption. Person-to-person transmission represents another critical route of Nipah virus spread, particularly in Bangladesh and India. Approximately one-third of cases in Bangladesh resulted from direct human-to-human transmission. The virus spreads through close contact with infected individuals, and numerous instances of nosocomial transmission in healthcare settings have been documented. Healthcare workers caring for Nipah patients face substantial risk if adequate infection prevention and control measures are not implemented. Family members and caregivers of sick individuals have also contracted the virus through close contact during caregiving activities. Since 2001, Bangladesh has experienced nearly annual outbreaks of Nipah virus. Through September 2025, Bangladesh's surveillance system had documented 347 cases with an overall case fatality rate of 71.7%. While the yearly number of reported infections has generally remained below 10 cases since 2016, with an exception in 2023, each outbreak represents lives lost and communities devastated. In 2025, Bangladesh reported 4 confirmed fatal cases between January and August, occurring at different times in 4 separate districts across 3 different geographical divisions. All cases were temporally and geographically unrelated, indicating multiple independent spillover events from the animal reservoir. India has experienced multiple outbreaks since an initial cluster in Siliguri, West Bengal in 2001. Kerala state has been particularly affected, reporting outbreaks regularly since 2018, with 9 separate outbreaks documented between 2018 and 2025. The 2018 Kerala outbreak proved especially deadly, with 23 confirmed and probable cases and a case fatality rate of 91%. All cases were traced to a single index patient, with transmission occurring across 3 hospitals through primary and secondary cases. Between May and July 2025, Kerala reported 4 confirmed cases, including 2 deaths. Most recently, in January 2026, 2 epidemiologically linked cases were confirmed in West Bengal state, involving 2 nurses working in the same hospital in Barasat, near Kolkata. Both had developed symptoms in late December 2025 and were confirmed positive on January 13, 2026. The Philippines experienced a single outbreak in 2014 that revealed yet another transmission route. 17 cases occurred in 2 villages, with 9 deaths, resulting in a case fatality rate of 52.9% among those with acute encephalitis syndrome. Investigation linked the outbreak to the slaughter and consumption of horse meat from horses that had recently died. Some cases involved human-to-human transmission among healthcare workers, and even domestic animals that consumed the horse meat became ill and died, including 4 cats and 1 dog. No further outbreaks have been reported from the Philippines since 2014. Chapter 5: Current Diagnostic Methods and Surveillance Systems. Accurate and rapid diagnosis of Nipah virus infection presents significant challenges because the initial symptoms resemble many other infectious diseases. Fever, headache, and respiratory symptoms could indicate influenza, dengue, malaria, or numerous other conditions common in South and Southeast Asia. Similarly, encephalitis symptoms could result from Japanese encephalitis, herpes simplex virus, or other encephalitic pathogens. Laboratory testing is essential to definitively diagnose Nipah virus infection. Real-time reverse transcription polymerase chain reaction testing serves as the gold standard for diagnosing acute Nipah virus infection. RT-PCR can detect viral RNA in respiratory samples, blood, or cerebrospinal fluid obtained through spinal tap. The test provides rapid results and high sensitivity during the acute phase of illness. However, the timing of sample collection matters significantly because viral loads change throughout the course of infection. Serological testing detects antibodies produced by the immune system in response to Nipah virus infection. Enzyme-linked immunosorbent assay can identify IgM antibodies, which appear during acute infection, and IgG antibodies, which indicate past infection or convalescence. ELISA testing proved valuable during the initial Malaysian outbreak, with IgM detection from cerebrospinal fluid or serum being common among confirmed cases. The development of Nipah virus-specific IgM and IgG ELISA protocols has improved diagnostic capabilities, particularly for retrospective outbreak investigation and seroprevalence studies. Plaque reduction neutralization tests provide confirmatory testing by measuring functional neutralizing antibodies, while highly specific. These tests require specialized biosafety level 4 laboratories and are more time-consuming than other methods. All samples from suspected Nipah virus patients must be handled with extreme caution as they represent a serious biohazard. Laboratory testing on non-inactivated samples requires maximum biological containment conditions, and only trained personnel working in suitably equipped laboratories should process these specimens. Bangladesh has developed a comprehensive surveillance system to detect and respond to Nipah virus outbreaks promptly. Established in 2006, the National Nipah Surveillance System has documented all 347 cases reported through September 2025. The system integrates multiple approaches, including hospital-based surveillance at sentinel sites, event-based reporting from healthcare facilities, and community engagement to identify suspected cases. An innovative development in Bangladesh's surveillance strategy involves post-mortem surveillance implemented in 2023. This approach recognizes that many Nipah deaths occur before diagnosis, particularly in rural areas where healthcare access is limited. By systematically investigating deaths that meet specific criteria for possible Nipah infection, the surveillance system can detect cases that might otherwise go unrecognized and provide more accurate estimates of disease burden. Contact tracing forms a critical component of outbreak response. When a case is confirmed, public health teams work rapidly to identify all individuals who had contact with the patient during their infectious period. Close contacts undergo a 42-day follow-up period with monitoring for symptoms. During the 2025 Bangladesh outbreaks, contact tracing identified 96 contacts for the first case and 71 contacts for the second case. Fortunately, all tested negative for Nipah virus. This intensive surveillance of contacts allows for early detection of secondary cases and helps interrupt transmission chains. Chapter 6: Current status, prevention efforts, and future directions. As of January 2026, Nipah virus continues to pose a persistent threat in South and Southeast Asia. The most recent confirmed outbreaks demonstrate the virus's ongoing epidemic potential. Between January and August 2025, Bangladesh reported 4 fatal cases from unrelated spillover events in different districts. India documented 4 cases with 2 deaths in Kerala state between May and July 2025. Most recently, West Bengal state reported 2 epidemiologically linked cases among nurses in January 2026, with both cases confirmed on January 13. While one nurse has begun recovering, the other remains in critical condition as of late January 2026. The World Health Organization assesses the overall public health risk posed by Nipah virus at the national and regional levels to be moderate, with the risk of international disease spread considered low. However, several factors contribute to ongoing concern. The ubiquitous nature of fruit bat reservoir hosts, increasing deforestation that brings bats into closer contact with human populations, multiple modes of transmission, including human-to-human spread, the extremely high case fatality rate, and the lack of licensed treatments or vaccines. Prevention efforts have focused heavily on non-pharmacologic interventions to reduce human exposure to the virus. Physical barriers to prevent bat contamination of date palm sap have shown promising results. Interventions include covering sap collection pots with bamboo skirts, jute sticks, or polythene barriers that prevent bats from accessing the collecting areas while still allowing sap to flow. Studies demonstrated these physical barriers could effectively reduce contamination. Public health education campaigns emphasize the importance of boiling date palm sap to molasses rather than consuming it raw, and thoroughly washing and peeling fruits before consumption, especially discarding any fruits showing signs of bat bites. In healthcare settings, strict infection prevention and control measures are essential. The WHO recommends standard precautions at all times for all patients, with enhanced measures for suspected or confirmed Nipah cases. Patients should be placed in single-patient rooms, and healthcare workers should use contact and droplet precautions, including well-fitting medical masks, eye protection, fluid-resistant gowns, and examination gloves. During aerosol-generating procedures, airborne precautions are necessary, including airborne infection isolation rooms and fit-tested filtering facepiece respirators. Currently, no licensed vaccine or specific antiviral treatment exists for Nipah virus infection. Treatment remains supportive, focusing on managing complications, providing oxygen therapy when needed, applying specific organ support such as mechanical ventilation or renal dialysis, and ensuring adequate hydration and nutrition with frequent monitoring. Early diagnosis promotes early supportive care, which can prevent deaths from severe viral infections. Ribavirin, a guanosine analog with broad-spectrum antiviral activity, was used during the Malaysian outbreak with mixed results, and current Indian guidelines recommend it for treatment, but not for post-exposure prophylaxis. Multiple promising therapeutic and vaccine candidates are in development. The human monoclonal antibody M1024 targets the ephrin B2 and B3 receptor binding sites on the Nipah virus G glycoprotein. Studies in ferrets and African green monkeys demonstrated that M1024 provided complete protection as post-exposure prophylaxis when administered 10 hours after lethal Nipah virus challenge. This represents the first successful post-exposure passive antibody therapy for Nipah virus using a human monoclonal antibody. The antibody has advanced to phase 1 clinical trials. On the vaccine front, major progress is occurring in Bangladesh, the country most affected by recurring outbreaks. The PHV-02 vaccine candidate, developed by Public Health Vaccines and funded by the Coalition for Epidemic Preparedness Innovations, is entering mid-stage human trials in early 2026. This vaccine uses a live, attenuated recombinant vesicular stomatitis virus vector, expressing both the Nipah virus glycoprotein from the Bangladesh strain and the Ebola virus glycoprotein. The dual expression means PHV-02 could potentially provide protection against both Nipah and Ebola viruses. The technology is based on the licensed Ebola vaccine RVSV-ZBOV, which successfully combated previous Ebola outbreaks. Another vaccine candidate, CH-Ad-OX1 Nipah B, uses a replication-deficient simian adenovirus vector encoding Nipah virus Bangladesh strain glycoprotein G. Preclinical studies in Syrian hamsters showed that all vaccinated animals survived lethal challenge with Nipah virus Bangladesh strain without viral RNA detection in oropharyngeal swabs or tissues at necropsy, while control animals died. The vaccine is also moving toward clinical trials in Bangladesh. Scientists continue investigating multiple other antiviral agents. Remdesivir, an adenosine nucleoside analog with broad-spectrum activity against paramyxoviruses, coronaviruses, and filoviruses, has demonstrated in vitro activity against Nipah virus and improved mortality in non-human primates. Favipiravir, a purine analog RNA-dependent RNA polymerase inhibitor approved in Japan for influenza, showed activity against henipaviruses both in vitro and in Syrian hamster models. The fact that Nipah virus has an RNA genome means it can mutate rapidly, potentially developing resistance to antiviral drugs or evading vaccine-induced immunity. Research indicates the Bangladesh strain shows increased intrastrain genetic variability compared to the Malaysian strain. This genetic plasticity represents a challenge for therapeutic development, but also underscores the importance of pursuing multiple approaches simultaneously. Climate change adds another dimension to the Nipah virus threat. Shifting habitats are bringing fruit bat populations into closer contact with human populations across South and Southeast Asia, potentially expanding the geographic range of spillover events. An estimated 2 billion people live in areas where they could potentially be exposed to Nipah virus. The sporadic and unpredictable nature of Nipah virus outbreaks creates challenges for traditional clinical development pathways. With cases numbering in the single digits most years, conducting large randomized controlled trials becomes logistically difficult. Scientists have suggested using vaccine candidates on a compassionate basis to prevent infection in exposed individuals, which could provide real-world effectiveness data while potentially saving lives. WHO has designated Nipah virus as a priority disease under the WHO Research and Development Blueprint, recognizing its epidemic potential and the urgent need for medical countermeasures. International collaboration between at-risk countries, research institutions, funding agencies like CEPI, and public health organizations continues to drive progress toward effective diagnostics, therapeutics, and vaccines. Continued vigilance through robust surveillance systems, early case detection, rapid outbreak response, strict infection prevention and control in healthcare settings, public education about risk factors and prevention measures, and sustained investment in research and development represent humanity's best defense against this deadly pathogen. The scientists working on Nipah virus tell us that while the challenges are substantial, the tools to combat this virus are within reach. The PHV02 and CH Ad-OX1 Nipah B vaccines entering human trials in 2026 may finally provide the protective shield that vulnerable populations desperately need. We stand at a critical juncture. The knowledge accumulated since that first outbreak in Malaysia nearly 3 decades ago has revealed the virus's secrets, its bat reservoir, its transmission routes, its devastating pathology, and its molecular machinery. Armed with this understanding, the scientific community is developing increasingly sophisticated countermeasures. Success will require sustained commitment, adequate funding, international cooperation, and the courage of trial participants in outbreak-affected regions willing to test experimental vaccines and treatments. If we rise to meet this challenge, the annual tragedies that unfold in Bangladesh and India may finally come to an end. The virus that emerged without warning in 1998 need not continue claiming lives indefinitely. Science offers hope, and that hope grows stronger with each passing year.